Okay, hello guys, I'm Oliver. Today we are going to talk about B1.2 proteins in biology. So first, proteins is a macromolecule, meaning that it has carbon in it, and also it is made of amino acids, which is the monomers of proteins. There are 20 different types of acids. Yeah. So first, this is the basic chemical structure of amino acids. We need to memorize this. And yes. The R group is what gives each amino acids its properties. It is what makes them different. So every R group is different. Uh, Amino acids, they break or they bond to each other with hydrolysis and condensation. So they bond using condensation. So what happens is uh, this hydroxyl group and the hydrogen atom comes out to produce oxygen, to produce water, and then they will combine to each other just like this. And to break down, hydrolysis will happen, where water will be used, and then uh, it will make it like this. Yes. So if a lot of amino acids bond to each other, they form peptides. Yes, this is also called peptide bonds. And if there are a lot of amino acids, then we call it polypeptide. Essential amino acids and non-essential. Essential means that we can produce it in our body. Essential means we cannot produce in our body, so we need to eat food to get that amino acid. And non-essential amino acids means that we can produce it in our body. Polypeptide. So it is made of peptide bond, which is this, and they can form a long chain. And some can contain several hundred amino acids, which is very long. And a polypeptide can function by itself, but it can also bond with another polypeptide to function. So DNA are codes of sequence of amino acids. So you need 3 codons or nucleotides for each amino acid. And if you have, for example, 400 amino acids, then you will have about 1,200 codons or nucleotides. And you also have codes before and after reading frame, and also non-translating codes. And because we have 20 types of amino acids, and that can form a lot of combinations, so this is why DNA sequences can be very different, and it also makes uh, every organism's different. Number of polypeptides. So, for example, lysozyme, which is a type of enzymes that break down things in your cells, it only has one polypeptide. It is made of one. And integrin is membrane proteins, so things can travel through membranes, and it is made of 2 polypeptides. Collagens, which is a type of structural proteins, is made of 3, and hemoglobin is a transport proteins in your blood to carry oxygen, and it is made of 4 polypeptides. So about protein denaturing. Denaturing is when your proteins cannot function anymore because the protein's 3D structure is lost. So this is because of intramolecular, no, intermolecular uh, forces, not the peptide bond. So it is like the hydrogen bond uh, that is breaking, not the bonds that is here. This is intramolecular. So because the bonds that maintain protein structure are altered permanently, so the protein denatures and it can't be used anymore. And these bonds are weak. They can be changed when you have more extreme temperature or pH level that the protein doesn't function well in. So that is it for the SL content. And now for HLs, um, we need to know that the R groups determine the properties of uh, of the protein. So you have two types, hydrophobic and hydrophilic. Hydrophobic is just non-polar. Um, so equal distribution of charge, and hydrophilic can be polar, they can be charged, they can be acidic, and they can also be basic. Primary structures. So primary structure is basically the sequence of amino acids, and this is important because every sequence means that you have different R groups, and when they interact with each other, they can form different shapes. So the sequence of the amino acids determine the shape of the protein. So primary structure cannot stand alone because uh, there will be other interactions. So further foldings like secondary structure. So the polypeptide chains will form either alpha helixes and or beta pleated sheets. These are two common types, and this is because of the hydrogen bonding to stabilize the structure in the backbone. So the backbone is um, these two, these three, and not the R groups. We can see that uh, uh, alpha helixes are made because H is bond with O in this shape, and in beta pleated sheets they bond with the one nearby them. And we need to recognize these two and differentiate them. Tertiary structure is the further folding of polypeptide, but this time it is R groups. So it can be hydrogen bonds where polar R groups are bonded to each other because if they're both polar, then they will attract to each other. Ionic bonds is because the R groups, amine groups, and the carboxyl groups can be charged by taking like hydrogen, and then they will attract opposite charged R groups. Hydrophobic will, which is nonpolar, will uh, bond with other hydrophobic amino acids. And this is another one we need to know, which is disulfide covalent bond, which is cytosines, which have sulfur atoms, and sulfur will bond together. So during tertiary structure, the hydrophilic, which is the polar part, will go outside of the cell, not cell, the protein, because it is hydrophilic, so it can attract water and it will protect the hydrophobic nonpolar in the core, so they won't have to interact with water. Quaternary structure is when you have two or more polypeptide chains attracted to each other and to make a functional protein. This is because of intermolecular forces like hydrogen bonding. We have here hydrophobic, ionic, and more. There are two types, non-conjugated and conjugated. Non-conjugated means that you have only proteins that is making the polypeptide, no, the protein. For example, you have insulin and collagen. And conjugated means that it has non-protein component. An example is hemoglobin, because it has four heme groups, and each it has iron in it. So this is why we have iron in our blood, and it is used to take in oxygen, and so oxygen will travel in the bloodstream and carry to the organs. The last thing is that we have two types, fibrous and globular. This one means that it is simple and composed of long and narrow strands. It has structural roles. So like collagen, this is a fibrous proteins. It is stable, strong, and like collagen makes the skin's tendons and bones. Globular proteins are more complex. It is compact, it is round, and has a functional role. For example, enzymes are made of globular proteins. They are soluble in water because uh, the outside is made of hydrophilic and inside is hydrophobic to maintain the structure. And also, insulin is an example of globular proteins. Yeah, so that is it for 1.2 proteins.